अस्सलाम वालेकुम स्टूडेंट। आज हम अपनी न्यू बुक के लिहाज से इसमें जो एक्सपेरिमेंट नंबर 3 है और 4 जो कि क्रोमेटोग्राफी से रिलेटेड है, इसको हम देखते हैं कि ये क्रोमेटोग्राफी क्या है, क्यों परफॉर्म करते हैं और कैसे परफॉर्म की जाती है। और स्टूडेंट्स यकीनन जो है ऑलरेडी अपने कॉलेजेस में ये परफॉर्म कर भी चुके हैं। स्कूल लेवल पे भी अगर कोई कैम्ब्रिज सिस्टम से या खान बोर्ड से आया है तो वो ऑलरेडी उसको परफॉर्म कर भी चुका है। तो देखिए, सबसे पहले तो ये समझ लीजिए कि लव्स क्रोमेटोग्राफी, दिस इज द इंग्लिश वर्ड क्रोमेटोग्राफी। व्हाट डज इट मीन? क्रोमेटोग्राफी। क्रोम कहते हैं, एक्चुअली क्रोम मीन्स colored. This is the Greek word chroma, and from the same word in biology you have used the term chromosome, because when after studying he observed the cell he found, and the scientist, the botanist, found the chromosomes. So that's why they are called chromosomes, means colored bodies. So chroma means colored. Graph, graph means to distribute or to write. To distribute. अब जैसे आप जानते हैं जियोग्राफी, distribution of different types of land all over the world, all over the globe. So graphy means distribution, to distribute or to write. आप अक्सर ग्राफ प्लॉट कर रहे होते हैं डिफरेंट एक्सपेरिमेंट्स में, डिफरेंट थियोरेटिकल टॉपिक्स के अंदर आपने ग्राफ देखे। सो what what do you do there? You actually distribute your data on the page. So graphy means distribution. So chromatography means distribution of the colors. But we know that it is not always the distribution of colors. But actually, the first scientist who discovered it, he was not chemist, he was the botanist. Botanist means Mahir Nabatiyat, who is the expert of studying plants. So he actually separated different plant pigments by using the chromatography technique first time, and the name was sweat. T is silent here. So he first introduced chromatography and he separated the colors or the pigments of the plant. That's why the name is derived chromatography. But today we can separate the colorless substances as well. For example, different types of sugars, different types of amino acids. And here in our syllabus, in experiment number 3, the object is this, to separate given mixture of ink by paper chromatography. Now the word paper. There are different types of chromatography: column chromatography, paper chromatography, high performance liquid chromatography, TLC, thin layer chromatography. So this is not the time to describe everything in detail. We just focus on our experiment. So what we need in uh, our experiment, in this experiment, we need a chromatographic tank. This is the chromatographic tank. It usually it is it is rectangular in shape, but it could be uh, of different shapes and different sizes, depends on the size of your chromatogram that you will use. For example, here, this is a rectangular chromatographic tank, and here there is a solvent. The solvent could be water, it could be the mixture of two different organic solvent, or the organic solvent with water. So the first is the TLC tank. Second, if you are performing paper chromatography, then the Whatman filter paper strip is required. Why Whatman filter paper? Because it has a specific pore size and specific properties that are good for paper chromatography. So we use a paper, and on the paper, what we do, we put a spot that I will show you. And for putting a spot, that is in technical term called loading of the sample. For example, this is the strip of the Whatman filter paper. It should be uniformly, it should have uniform width. So there is a origin line, and we load our sample of ink here, because here we are doing paper chromatography for separating the mixture of ink. So the sample is loaded here. And for loading, we need capillary. See, this is you can consider it as your paper. So we are loading through this capillary, the spot over here. This capillary can be used for other material as well, but right now our object is to separate the ink, to separate the component of the ink. So here we are loading the sample, loading sample. And here it is chromatographic tank. And in the tank, there is a small quantity of solvent. Solvent is there. And this solvent can have different composition. Okay, now go to the previous page. And we need we need a thread, not always. If your paper can be put easily here, then no problem. You do not need thread. But if paper cannot be put here, so we need papers to connect it, or to press it under the lid of the TLC tank, so that um, it can be kept in proper position. Pencil, why we need pencil? Pencil is needed to draw this line. We always draw the line with pencil, never we use pen, because pen itself has component that can be separated by any solvent that can spread the ink of the pen. That's why we use graphite pencil. So the content of the pencil cannot be move up by any solvent. So we always use the pencil. Ruler for making this straight line, as well as for further measurement after the development of chromatogram. And watch glass. Chemical, in this chromatography, they have used methanol and distilled water as a solvent. In certain ratio, they have made this solvent. So in this diagram, you can consider that this solvent is methanol plus water, and we use the distilled water to avoid any discontamination. Distilled water. Okay, now see the theory. Paper chromatography is an experimental technique for separating dissolved substances based on their different travel rate across the paper, across the stationary phase. In chromatography, there are two important terminologies. One is the stationary phase. And remember that in the spelling of stationary, there is ARY in the end, not ERY, because that stationary is your pencil, pencil pen and other things. So this is the stationary phase. And another one is the mobile phase. Stationary phase does not move. And mobile phase, usually in general chromatography, it moves up. And mobile phase is actually solvent, while mobile phase is solvent. But the stationary phase could be different. Okay. So what is the function of these stationary phase and the mobile phase? Actually, the component that we are going to separate have interaction with both the mobile phase and the stationary phase. Some has more attraction or more affinity towards the stationary phase. So stationary phase here in your book, it is written that the paper surveys stationary phase. But actually, this is not the stationary phase. I don't know why they have made this mistake, because in paper chromatography, stationary phase is actually water molecules with the Whatman filter paper. Whatman filter paper is made up of cellulose. So along the cellulose fiber, suppose these are the cellulose fiber in crisscross arrangement. So in the paper, there are certain water molecules. So these water molecules which do not move and it remain over there bonded with the paper, it serves as stationary phase. Okay. So this is the mistake in your book, that paper serve as stationary phase. This is not the true case. I will show the reference of the book as well, the proper uh, authentic books of analytical chemistry, that paper does not serve as a stationary phase, but the water, bound water on the paper is serve as a stationary phase. So here, this is the mixture of methanol and water. This is also liquid, and the bound water is also liquid. So that's why paper chromatography is a kind of liquid chromatography, where both the stationary and both the stationary and the mobile phase are in liquid state. So that's why it is called liquid chromatography. In the end, I will show the reference that what is the proof that I'm telling that paper itself is not the stationary phase, but the water bound in the paper is the stationary phase. So you should you must be very clear about it, because in higher studies you will perform chromatography if you will keep chemistry in your as a subject. Okay, so there are two things: stationary phase and the mobile phase, and during so the mobile phase is the mixture of methanol and water. In this experiment, dissolved pigments of ink move up with the mobile phase along the paper strip. As you can see here, these are moving up. The ratio of the distance traveled by the component of mixture to the distance traveled by the solvent is known as retention factor. And shortly it is written as RF. So we calculate the retention factor. What is the retention factor? Distance traveled by the component of the mixture. Different component have different RF values. And distance traveled by the solvent, and up to what solvent moves up. I will show a video that is similar to the paper chromatography, but it is uh, not for the separation of ink, but it was separation for plant pigments. Okay, so this is the theory. So you must write the theory for this minor experiment in your first year chemistry practicals. It has 1 mark. So you have to explain in your own words to cover all the important terminologies here. And as it is written in your book that paper survey stationary phase, so there is no harm if you write the same sentence. But being a teacher, this is my duty to describe you the correct, to tell you the correct information, and to correct any mistake in our books or the literature. Okay, and in the end I will show the proof of this concept. Okay, now the procedure, as I have told you, that what we do first, we make a mixture of methanol and distilled water as per requirement. So it is in your book, it is written 3:1. You can see here paper solvent system by mixing 15 ml methanol into 5 ml distilled water. So 20 ml is total solvent there. So the ratio of methanol to distilled water is 3:1 because 15 ml methanol can be represented by this is to water H2O. So this is 15 ml is to 5 ml. So simplified ratio is 3:1. So the total 20 ml solvent is taken in the tank, in the chromatographic tank. How much volume of the solvent should be taken? It depends on the size of the TLC tank. Okay. Now, next. And they have also already mentioned that volume of the mobile phase may vary depending on the availability of the chromatographic tank. If the chromatographic tank is larger, so definitely you need little more volume of the solvent, means the mobile phase. So this is enough to write up to here. Second step, pour the solvent system into the chromatographic tank and cover it by tank lid. In order to avoid the evaporation of solvent and maintain the internal atmosphere. What does it mean? So actually what we do, first we add the solvent here and we cover it, and we must leave it for some time so that the vapors uniformly saturate the atmosphere inside the chromatographic tank. So for the solvent system is mobile phase. You can use the term solvent system as well. into chromatographic tank and cover it by tank lid in order to avoid the evaporation of the solvent and maintain the internal environment. So another important thing is that if you not cover, methanol is more volatile than water, so methanol can evaporate. So the ratio will be disturbed. And one more thing, maybe during the chromatography, your solvent evaporates. So how can you determine that how long the spots will run? Next, put few drops of ink, red ink, or any other ink that is provided to you. It could be black ink, and a few drops of blue ink into the watch glass, and then mix them using a glass rod. So you are already mixing two non-components, but you can select uh, directly the black ink. Black ink comprises three different colors. So all the components can be separated. But here in this method, they have given you red and blue ink. So in the glass rod, you have mixed them together. Then using pencil, draw a line. This line was drawn by the pencil. Okay. So using pencil and scale, draw a baseline that is called the baseline. Use these important terms, origin line or the baseline on the strip of the filter paper 2 centimeter above. Why it could be less than 2 centimeter or above 2 centimeter? Because when you put it into the TLC tank, this baseline must be above the level of the solvent. It should not dip into the solvent. This line must be well above the upper level of the solvent. Got it? So that's why we do this. Place 1 drop of ink mixture on the baseline of the filter paper strip using the capillary tube, as I'm doing here, just like this. Since the solvent is mobile phase, sorry, suspend the strip of Whatman filter paper into the chromatographic tank by using thread in such a way that the lower end of the strip is dipped 1 centimeter, not accurately, but uh, just an idea, into the solvent, but it does not touch the baseline. The same thing that I told you, that this baseline should not touch the solvent. It should not be under the solvent. Okay. So it must be 1 centimeter above the solvent, but not necessarily 1 centimeter. It could be zero 0.1 centimeter. No problem. So, but in our method, it is written that 1 centimeter above. Now close the tank by putting tank lid. So the tank lid, after putting this lid, would be over the tank again. And the same reason that solvent should not be evaporate and saturation should be kept constant in the tank. Since the solvent is a mobile phase, and here the solvent is the 3:1 mixture of methanol and water. It rises slowly along the pigments of the ink. The ink spot separates into two colored bands, red and blue. So the diagram is little bit disturbed. So there is a blue spot and there is a red spot. Okay. When the solvent has risen on solvent front line, there are two ways to get the solvent front line. One is that when you are developing this chromatogram, then you draw the line over here. Or otherwise, You don't do this. And you run it, and immediately when you keep it out, then there would be the wet portion in the chromatogram. Suppose this is the baseline and the solvent runs up to here. So up to here, you will see it is wet. So where it is wet, you draw the line. Okay. But you should draw it immediately, because again, if your solvent evaporates, then how will you find that where what was the correct solvent front? Why it is called solvent front? Because components traveled up to here or here, but the solvent traveled up to here. So the distance traveled from this line to this line is the solvent front. Means the distance traveled by the solvent. So here the formula, the distance traveled by the component of the mixture and divided by distance traveled by the solvent. So you can say that retention factor is, you can write here, solvent front. Okay. When the solvent has risen up to solvent front line, remove the paper strip from the tank. The paper strip is now called chromatogram. अभी जो आपका पेपर स्ट्रिप है, अब इसको क्रोमेटोग्राम कहेंगे क्योंकि इसके अंदर वो सेपरेशन हो चुकी है. Now mark the position of the center of blue and the red. For example, if this is the chromatogram and suppose this is the I think I should write. Okay, leave it as it is. So this is the baseline and this is the solvent. This is the solvent front and there is a red spot and there is a blue spot. This is the red spot and this is the blue spot. So what will you do after developing this chromatogram? You measure the solvent front. Your ruler must be very accurate to the line. If you will, अगर ये तिरछा होगा, then you will get the wrong result. And then you want to, 1 minute, you want to measure the solvent front for red spot. So the line must be at the center, means if this is the spot, then you will measure up to the center of the spot. Not here, not here. Okay. So it should be from the center. So that would be the distance traveled by the red spot and This is the distance traveled by the 1 minute. And this is the distance traveled by the blue spot. And you can consider it y. And this is the z. So the distance traveled by the by the component of any component of the mixture, this is called solute front. So RF can be written as solute front divided by the solvent front. And you remember that solvent front is always greater. So for red spot, you can calculate RF by X upon Z, while for blue spot, you can calculate by Y upon Z. So now you will see how much it will travel. So whatever the answer, it must be less than 1. Remember that RF is always less than 1. और अगर ये बहुत ज्यादा सॉल्युबल है तो फिर ये हो सकता है 1 भी हो जाए, तो फिर वो सेपरेशन तो ना हुई। उसके बारे में हम श्योर नहीं है कि फिर वो सेपरेशन हुई कि नहीं हुई। तो आरएफ की वैल्यू, क्योंकि देखिए सॉल्वेंट हमेशा उसकी वो ज्यादा ट्रेवल करेगा एज कम्पेयर टू सॉल्यूट, जो भी आपके कंपोनेंट्स होंगे। यहां पर आपने इस एक्सपेरिमेंट में सपोज करें, हम यहाँ पे हाइपोथेटिकल वैल्यूज रख लेते हैं। डिस्टेंस ट्रेवल्ड बाय द इंक, डिस्टेंस ट्रेवल्ड बाय द सॉल्वेंट। अच्छा, ब्लू एंड रेड। सो सपोज ब्लू ट्रेवल अप टू 1 centimeter, blue has traveled 1 centimeter. Centimeter is already written over there, so you don't need to rewrite. This is the rule for writing something in the table. And it travels up to 3 centimeter. And definitely solvent front is same for both of them. And it was 5 centimeter. So the RF value for red one, I have taken it X and it is as Y. So it is 3 upon 5 and it is 1 upon 5. So 1 upon 5 is, what should be it? 0.2. You can use your calculator as well. And here it is 3. So what is this? So 5, 6 are 30. So this is 0.6. The RF value for blue one is 0.2. And RF value for red is 0.6. So the given mixture consists of red and blue ink. We have seen it. And the RF value of red ink is 0.6. And for blue ink, it is 0.2. So this is the way to carry out this, to conduct this experiment and to calculate the RF values. Okay. So in the examination, you have 1 mark for the observation. Up to this is observation, and you have calculated the value, and finally you get the result. So 1 mark is for result. So this is the minor practical of 3 marks, and it is not time consuming. So you can conduct this experiment in the start of your practical. So when you go to the practical day, first you perform this minor experiment. Now I'm going to show you a simple video of chromatography. It is not the paper chromatography, but somewhat similar. So watch the video. अच्छा, अब देखिए यहाँ पर ये हमारे पास ये एक टीएलसी टैंक। तो ये जो हमारे पास टीएलसी टैंक है, अब ये वीडियो रन होगी तो आपको मालूम होगा यहाँ पे इसमें सॉल्वेंट भी है। अच्छा, ये जो मैंने एक्सपेरिमेंट परफॉर्म किया है, इट इज नॉट द ink separation of the ink, but it is the separation of the plant pigments from the spinach leaves. पालक के जो पत्ते होते हैं उनमें से ये सेपरेट किया गया है। अभी मैं आपको स्टेप बाय स्टेप बताते जाऊंगा। सो सी हियर, आई एम स्टार्टिंग द वीडियो। देखें हमने क्या किया, ऊपर से लिड को रिमूव किया, और लिड को रिमूव करने के बाद हमने यहाँ पर ये 3 स्पॉट लगा हुआ क्रोमेटिक, एक क्रोमेटोग्राफिक प्लेट जो है वो उसमें हमने एंटर कर दिया। दिस नॉट द पेपर, दिस इज समथिंग एल्स, बट somehow it works like the paper. So here this is the spot from the leaf, and this is the spot from the twig. Means जैसे कि for example this is the spinach leaf. So in the spinach leaf, what I did, I cut the twig. This twig has been cut, and this part was, so twig has been removed from there. So this was the twig. I was comparing the pigments इन द स्पिनच एक्चुअली, लेकिन ये आपका काम नहीं है। आप तो सिंपल ये देखना है, ये वीडियो दिखाने का जो मेरा मकसद है, माई एम इज टू मेक इट विजुअलाइज कि क्रोमेटोग्राफी हो कैसे रही है। तो अब आप यहाँ पे देखिए कि यहाँ पर आपको इससे गर्ज नहीं होनी चाहिए कि व्हाट वाज द पर्पस ऑफ द एक्सपेरिमेंट, क्योंकि यहाँ पर ये आपके लेवल से अबव है। यहाँ पर आप सिर्फ ये देखिए कि ये किस तरह से क्रोमेटोग्राफी परफॉर्म हो रही है। तो आप देखिए सबसे पहले हमने क्या किया, वी हैव put the solvent. There is a solvent in the TLC tank, in this chromatographic tank, and we have covered it for some time to get the saturation of the vapors. And here the mixture, and what was the mobile phase? It is HA. HA is actually, it does not contain water. H is hexane. Hexane is a liquid hydrocarbon, and A is the acetone, and ratio is 3:2. And for this TLC tank, I got 10 centimeter cube of the total solvent. So how can you get this? If it is total 10 centimeter, it means I 6 ml 6 ml of hexane and 4 ml of acetone. So the simple ratio that that will appear is 3:2. So I use this solvent, okay, and no water is there. Anyway, so you can use different types of solvent depending on your experiment. So here I have used 3:2 mobile phase, that is hexane and acetone. And we have spotted, we have loaded the content of the twig, content of this is the mixture of twig and the leaves, and this is the component of the leaves only. Now see, that was my mistake, that it disturbed, and because of this disturbance, some of the solvent come over here already. It shouldn't be there, okay? But anyway, now see, now we have put the lid again, and see, solvent is running up, and with the mobile phase as it run up, separation has been started. You can see different bands: yellow band, green band, light green band. Now from here, you see it is moving faster. Actually इतना फास्ट क्रोमेटोग्राफी नहीं होती है, ये मैंने वीडियो को फास्ट किया यहाँ से, okay? So you can see the separation of the different colors in the leaf, in the twig. See, so if we have run it until up till now, so अगर हम इसको यहीं पर इसको निकाल देंगे, तो ये हो जाएगा आपका सॉल्वेंट फ्रंट, यानी बेस लाइन से लेकर यहाँ तक का जो डिस्टेंस है सॉल्वेंट फ्रंट। अगर मैं यहीं पे इसको निकाल लूँ, अगर मैं थोड़ा और रन करने दूंगा, तो फिर बाद में जो है इसका सॉल्वेंट फ्रंट आएगा। लेकिन अगर मैं सपोज करें यहीं से निकाल देता, तो ये सॉल्वेंट फ्रंट आ गया। फिर ये जो स्पॉट है इसके सेंटर से लेकर बेस लाइन तक, ये इस स्पॉट के लिए सॉल्यूट फ्रंट हो जाएगा। फिर ये इस ये जो ब्लू कलर का, ये ब्लू है एक्चुअली, यहाँ पे इतना वाजे नहीं है, तो इस ब्लू कलर के स्पॉट के लिए जो है फिर हम ले सकते हैं, इस येलो स्पॉट के लिए हम ले सकते हैं, यहाँ पे लाइट ग्रीन स्पॉट है इसके लिए ले सकते हैं। ठीक है, मैं आपको एक इमेज और दिखाता हूँ, इसकी क्रोमेटोग्राम की इमेज मेरे पास मौजूद है। आई एम गोइंग टू शो दिस। सो up to certain limit we stop our chromatography and immediately it is taken out, और फिर हम उसकी कैलकुलेशन कर लेते हैं। यहाँ पर एडवांटेज क्या है, आपने जो इंक क्रोमेटोग्राफी अपने कॉलेज में परफॉर्म की, और जो यहाँ पर प्लांट पिगमेंट क्रोमेटोग्राफी, इसमें क्या आपको नजर आ रहा है सब कुछ सेपरेट होता हुआ, because these are the colored. Allah has provided us perception in our eyes to observe these colors. हम इस कलर बैंड में से देख सकते हैं, तो ये नजर आ रहा है। लेकिन अगर ये अमाइनो एसिड्स या कोई और मटेरियल होता तो हमें नजर थोड़ी आ रहा होता, वो तो हमसे सॉल्वेंट फ्रंट लेते हैं, उसके बाद हमें कोई विजुअलाइजिंग यानी एजेंट यूज करना होता है जिससे हम उसको विजुअलाइज करते हैं, दैट इज कॉल्ड द स्टैनिंग एजेंट, स्टैनिंग रिएजेंट। तो बहरहाल वो नेक्स्ट क्रोमेटोग्राफी में भी हम देख लेंगे इसको, ये इंक क्रोमेटोग्राफी हो यानी सेपरेशन ऑफ इंक हो, या फिर सेपरेशन ऑफ प्लांट पिगमेंट्स हो, तो ये तो कलर्ड होते हैं, इसके लिए कोई इशू नहीं है, ये आपको डायरेक्टली नजर आ रहे हैं। अच्छा, तो अब यहाँ पर आप ये देख सकते हैं कि यहाँ पर ये आपके पास ये है, ये सॉल्वेंट लाइन है, तो यहाँ से यहाँ तक जो है इसका डिस्टेंस, ये यहाँ से यहाँ तक सॉल्वेंट फ्रंट हो जाएगा, यानी डिस्टेंस ट्रेवल्ड बाय द सॉल्वेंट। और फिर बाकी आपको जो भी स्पॉट लेना है उसके लिहाज से उसके सेंटर से ले लेंगे। तो बहरहाल दैट वाज द एक्सपेरिमेंट नंबर 3, और मैंने आपको एडिशनली प्लांट पिगमेंट की सेपरेशन दिखा दी ताकि आप उसको विजुलाइज कर सकें। कि देखें इस तरह से भी सेपरेशन हो सकती है, वरना आपने जो कॉलेज में परफॉर्म किया है पेपर के ऊपर इस तरह की इतनी अच्छी सेपरेशन नहीं आती। यहाँ पे मैंने कोई और मटेरियल यूज किया है, किसी और किस्म की क्रोमेटोग्राफी है, मैं उसके डिटेल में नहीं जाना चाह रहा फिलहाल। अच्छा, अब जैसा कि मैंने आपसे कहा था एक्सपेरिमेंट के शुरू में कि यहाँ पर थ्योरी में जो लिखा हुआ है कि पेपर सर्व एज अ स्टेशनरी फेज, दिस इज रॉन्ग। तो आइए मैं इसकी करेक्शन आपको शो कर देता हूँ कि किस तरह से, कहाँ से मैंने ये बुक में देखा है। तो मैं वो बुक का रेफरेंस भी आपको दे देता हूँ। अच्छा, आप देखिए इस बुक में, ये बुक कौन सी है, अभी मैं इसका टाइटल पेज भी दिखा दूंगा। ये एनालिटिकल केमिस्ट्री एक बहुत ही मशहूर बुक है, मैं अभी फिलहाल इसका नाम नहीं बता रहा हूँ। जो हमारे केमिस्ट्री के टीचर्स हैं वो इस बुक से बखूबी वाकिफ हैं। पेपर chromatography, separation by paper chromatography are performed in the same way as those in the thin layer plates. The paper are manufactured from highly purified cellulose with close control over porosity and thickness. यानी एक, जैसे हमने कहा था ना वॉटमैन फिल्टर पेपर चाहिए, तो वो बड़ा ही हमारे लिए जो क्रोमेटोग्राफी के लिए रिक्वायर्ड पेपर है उसके लिहाज से वो सिंथेसाइज किया जाता है, बनाया जाता है, मैन्युफैक्चर किया जाता है। Such papers contain sufficient, now see, such papers contain sufficient adsorbed water to make the stationary phase aqueous. मैंने आपसे कहा था ना कि जो वहां पर वाटर जो सेलुलोज के साथ एडहियर हुआ हुआ है, एडजॉर्ब हुआ हुआ है, वो वाटर जो है बेसिकली जो है वो सॉल्वेंट, वो जो है आपके स्टेशनरी फेज के तौर पे वर्क कर रहा है। ठीक है, तो इस वजह से हम इसको कहते हैं लिक्विड लिक्विड क्रोमेटोग्राफी। And the other liquids can be made to displace the water, however thus providing a different type of stationary phase। यानी अगर आप कोई और सॉल्वेंट इसको रिमूव करके कोई दूसरा जो है उसमें यूज करना चाहते हैं कि वहां पे वो एडहियर हो जाए, तो वो भी जो है एज अ सॉल्वेंट यूज हो सकता है। मगर हमें बहुत ज्यादा उसमें डिटेल में जाने की जरूरत नहीं है, लेकिन आप सिर्फ यहां तक ही देख लीजिए। तो जो सेलुलोज है, सेलुलोज इटसेल्फ स्टेशनरी फेज के तौर पे वर्क नहीं कर रहा है। स्टेशनरी फेज जो है आपका वो सेलुलोज नहीं है, स्टेशनरी फेज उसमें एड्सॉर्ब हुआ हुआ वाटर है। ये कौन सी बुक है आप देख लीजिए, मैं इसका फर्स्ट पेज पे चला जाता हूँ। ये पेज नंबर, ये कंप्यूटर में तो 981 आ रहा है, लेकिन इस बुक का जो ये पेज नंबर है इसको हम देख लेते हैं, इस बुक में कौन सा पेज नंबर है ये, ताकि रेफरेंस के लिए अगर कोई देखना चाहे तो देख सकते हैं। यहाँ पे पेज नंबर नहीं है तो ऊपर होगा। यस, पेज नंबर 942 है ये, चैप्टर 34 पेज नंबर 942 में। यहाँ पर ये 34B3 जो है ये पेपर क्रोमेटोग्राफी को स्लाइटली एक्सप्लेन कर रहा है। ये बुक है, देख लीजिए, स्कूज फंडामेंटल्स ऑफ एनालिटिकल केमिस्ट्री, 9th एडिशन। ये एडिशन भी कोई बहुत पुराना नहीं है, कुछ सालों पुराना ही है ये। ठीक है, तो आप यहाँ पर इसमें देख सकते हैं। और पेपर क्रोमेटोग्राफी एक बहुत पुरानी टेक्निक है, अब तो नई क्रोमेटोग्राफी की जो बुक्स है, एनालिटिकल केमिस्ट्री की बुक है, उसमें पेपर क्रोमेटोग्राफी जो है बहुत ज्यादा डिटेल से एक्सप्लेन भी नहीं करते हैं। तो बहरहाल हम जो है वापस उसी पोजीशन पे चले जाते हैं ताकि आप दोबारा देख लीजिए इसको। पेज नंबर 942 देख लीजिए। तो पेपर कंटेन्स sufficient adsorb water to make the stationary phase aqueous। ओके,